the man, he's the myth, he's the legend, he's the scientist with all the answers to all the questions. It's time for another episode of Alex's Science Corner.
Oh, thank you. No, thank you. Please,
What's going on? Wide world of science, my friend.
First thing I'm going to talk about is researchers have used some new technologies to see if they can remove HIV from mice. Now, this is a first step kind of process. The tool that they're using is called CRISPR, and it's a gene editing tool that has been, within the biology community, it has been a major leap forward for doing gene editing and studying genetics. One of the things that they've done is with HIV, when it gets into your system, a lot of it goes dormant in your system. And HIV, when it infects your cells, it changes the genes inside of your cells to make more HIV virus. And HIV doesn't kill you directly. What it does is it causes your body's immune system to get so weakened that everything else is going to kill you. Like a common cold can kill you if you've been infected with HIV.
Well, and there's certain cancers, too, that are associated with, if you've got HIV, they started seeing these rare cancers.
It makes it easier because your immune system has been knocked out by HIV.
Right.
One of the things is they've got a way to keep the HIV in check. They use a drug cocktail of antiviral drugs. They don't knock out the HIV. They just suppress it so that it's not doing anything. But you have to be on those drugs for your whole life. What they've been able to do in mice is using the CRISPR can go in and edit out the changes that the HIV has done to cells and take those changes out of the cells. And using a second method, which is called laser art, and what it is they use nanocrystals with antiviral drugs that stay in the blood system and in the cells for a very long time. And as parts of the HIV virus become undormant to go attack cells, it neutralizes those particular HIV viruses. The combination of the two they found has a 30% success rate in mice. That sounds really low, but before that it was 0% success. Mm-hmm. So having 30% of the mice have been cured, this is a major leap forward. The next step after getting the mice cured is now to go on to primates and see if we can have this kind of success rate in primates. And after a few years, if that's working, then they can start human trials. But this is one of those things, it's promising. They're using two different technologies right now to try and neutralize AIDS and get it suppressed. One, the AIDS viruses that have affected the cells, it goes in and turns the cells back, removes the AIDS virus from the cells.
Makes it a normal cell again. Yeah.
In a way, yes. Yeah. And so it's hopefully the combination of the two is what seems to be working right now.
Yeah, and you did send us the article, and it mentioned a couple of other different options that they were going to try this on. But I was surprised they didn't mention Lyme disease, because that does the same thing.
Well, I mean, they're doing work
goes
on
dormant.
They're doing work on HIV. So when it comes to this kind of stuff, they've got to spend time on one disease.
Right.
There's different teams that work on that.
Right. And once they've got the process down, I'm sure.
Well, what will be is the guys who are working on Lyme disease could say, "Hey, that's a thing we might see if we can try."
Well, but I'm sure they're going to wait until they've got some success with AIDS to say, okay, so here's the process that we used. And
what'll happen is the guys who do the Lyme research, they'll have to decide if that's going to be a process that'll work for them.
Right, but
I'm
sure they'll talk to
well, it's like everything else. It's like they're going to read each other's papers.
Yeah.
But next story is about octopus arms, and researchers, marine biologists, have found that octopi are very incredibly smart creatures. They've been able to, they understand cans, jars, they know how to open cans. They've been able to escape from aquariums. Seriously, they've been able to escape from aquariums and get out through sewer systems back out to the ocean.
And they're delicious.
Yeah, well, I think they figured out that's why they want to try and escape. What they found is that the octopi, they have a brain bulge in their main body, but most of their brain functionality is actually around their arms. Their arms have incredibly detailed nervous systems, and they have a sensory ring around the body where the arms connect to the body, and the arms will actually talk to each other before they talk to the brain. So
that's what happens.
Go ahead.
I know you were winding up for that. And body parts talking on their own.
Thank you.
Yes,
Thank you.
yes.
And thank you for allowing me to do it, too.
Yes.
Okay, go on.
And you can say it's like your hands just going places where your brain didn't know they were going.
Yeah.
But that's what's happening with an octopus. There's the arms, they are tasting, they're sensing, they're moving around almost without the main brain in the body knowing what they're doing. The brain kind of says, we're doing this, and then the arms figure out how to do it. They sense whatever they're going to sense. They'll react to each other. So two arms will know what each other is doing, but the brain may not know that two arms are doing something until they send a message later on.
That's kind of weird, though.
Yeah. There's one picture, and it's not in the actual article, but I've seen this before, where the arms will sense food and start putting it into the octopus mouth, and the mouth doesn't know that there's food there. Because
it's like, wait, what?
Yeah.
Oh,
Yeah,
wait.
It's like somebody else trying to feed you.
Oh, yeah.
Yeah. Yeah.
That's really odd. That
Yeah,
really is.
and again, they have so many sensors. All those little suction cups,
Mm-hmm.
they're like tongues. So each of them taste, feel, sense. The arms have so many sensory organs that for them to be able to function properly, they have to have their own brains in each arm, and to coordinate with each other. The ring of sensory, it has actually more neurons in that ring than the brain of the octopus itself has. So brain,
actually,
the
it
arms
has
actually,
more than one brain when you're really talking brains.
They've got a lot of subbrains.
9
it's
brains.
Like,
yeah.
Yeah, it's got it's got a subbrain for each arm, and then a subbrain that controls the arms, and then the main brain for the octopus,
Itself. So that's 10.
yeah, 10-ish, you could say.
Yeah.
It's more distributed than what we have.
We'll have to pull up that video. I heard Brian Cox talk about octopus. I'll pull up the video.
Again, octopi, cuttlefish, incredibly intelligent creatures. And cuttlefish are incredibly cool, too, because of the way they can change. I mean, octopi can do this, too. They can change their texture and their colors because of the
environment they're in.
Well, they can sense their environment and then change their skin color and their texture.
As a camouflage.
Right. And there's some amazing pictures of cuttlefish. And cuttlefish have this ability to do this flash in color. So it looks like they've got they can sort of hypnotize their prey with these flashing colors.
Well, going back to the octopus for a second, can't they regrow too if they lose an appendage?
That I can't remember right now.
Yeah, they could regrow.
Someone should look that up.
Yeah, I think so.
All right, anything else to add?
No.
All right, if you have any questions about these stories or anything else science-related or Formula One related, feel free to call Alex right now at 201...
To what? What? What?
203. I don't know where that came from.
837.
99.
24.